Hi all. This is the third episode of my project on the speculative evolution of the planet Parabola b. In this episode we will look at how our basal two motor cellular creatures evolved 15 and 100 million years after their appearance. Okay, I'll start by looking at the evolution of convacum. Perhaps one of the groups of convacum will develop a method of obtaining water when the hole narrows, increasing the pressure at a certain point, and from it a stream of water will rush into areas with lower pressure. Perhaps this water will pass through the filter of the eyelashes, collecting organic matter on the eyelashes. Afterwards, to remove organically depleted water, another muscle cells can develop from below, which when the hole is narrowed, will shrink, and when the hole is expanded again, they will be sharply stretched and the water will be pushed out. Eventually, such organisms may develop an attached lifestyle and a mineral shell for greater attachment. And I will call these attached organisms spongiae, which is Latin for sponges. It is possible that one group will separate from the main line and begin to lead an active lifestyle using jet locomotion, and it is possible that certain cells can stretch more and others less in order to change the direction of water flow and control orientation. It is also possible that they will develop a depression in the front that contains cilia for filtering. I will call these creatures reactive amphiltra. Considering that the original convacum ate unicellular organisms, it is possible that some of the reactive amphiltra may develop a cavity into a full-fledged container where unicellular digestion will take place. And I will call these predators reactive ampredatora. We're done with the evolution of convacum. Let's move on to algae for a moment. Some of the photosynthetic mats began to form the first multicellular algae. They drift in the upper layers of water due to air containers and have a large square flat surface. I'll call them square algae. Now let's move on to the evolution of raduas. In short, they simply developed stinging cells and threads. Their diet now consists of single-celled photosynthetic mat algae. I'll call them phylum Radiale. Now about the reproduction of these species, it has become sexual. In the case of convacum, gametes are created by budding on cells in original containers and are later released along with a new stream of water. In the case of raduas, gametes spot at the ends of the filaments. And in square algae, they have a hole that leads to the area where gametes are budding. Gametes of all groups cannot be fertilized by the gamete of their own parent. Now let's move 100 million years from the point when the last video ended. Here we will consider only one form. I am most interested in the carnivorous version of the convacum line because they are capable of digestion. And so here is the diagram of this creature. We have 8 through gut, 4 simple eyes, 4 simple brains, a ring of muscles, 4 gamete-generating glands, 4 reactive zones, as well as skin and a large number of cells storing oxygen and lipids, etc. They can use a ring of muscle cells to change the direction of the back for maneuvering. They still move jerkily and can't really resist the current, so they are actually plankton. And their size is 10 centimeters. I will call these life forms Proavis carnivora. That's all for today. In the next episode we will see how Proavis carnivorus differentiates into different niches and becomes a nekton. Well, today I say goodbye to you. Adios.